So let us discuss about other mechanisms of cell death. So other mechanisms of cell death include necroptosis, pyroptosis and ferroptosis. There are three other mechanisms of cell death apart from necrosis and apoptosis. So let us see what is necroptosis. So as the name indicates, this is a combination, that is hybrid of necrosis and apoptosis, which shares some aspects of both necrosis and apoptosis. So morphologically it resembles necrosis, as both necroptosis and necrosis are characterized by loss of ATP, swelling of the cell and organelles, generation of reactive oxygen species and release of lysosomal enzymes, which results in rupture of plasma membrane. So this portion, so loss of ATP, swelling of the cell and organelles and generation of ROS and release of lysosomal enzymes leading to rupture of plasma membrane is a phenomenon that occurs in both necrosis and necroptosis. Next one, it is also triggered by signal transduction pathways that culminate in cell death. That is, this type of necroptosis is a programmed necrosis, otherwise programmed necrosis, because it is triggered by signal transduction pathways, which will occur in cell death, and this feature is similar to apoptosis. Next, but in contrast to apoptosis, the signals that lead to necroptosis will not activate. That is very important. Not activate caspase enzymes. That is not result in caspase activation, and hence this is a necroptosis is otherwise called as caspase independent programmed cell death. That is, there is a signal transduction taking place in both apoptosis and necroptosis, but the caspase enzymes are not activated in this phenomenon. Therefore, necroptosis is also called as caspase independent programmed cell death. So the process of necroptosis starts by the ligation of a receptor by its ligand. So ligation of TNFR1, that is tumor necrosis factor 1, is the most widely studied mode of necroptosis. Okay, so that is very important. Ligation of TNFR1, as we can see here, will result in activation of other reactions essential for necroptosis. Okay, next, there are two kinases. Necroptosis involves two kinases, that is receptor interacting protein RIPK1, receptor interacting protein kinase 1 and receptor interacting protein kinase 3. Okay, so ligation of TNFR1 will help recruit these kinases into a multi-protein complex, and RIPK3 will phosphorylate a cytoplasmic protein called MLKL. So due to the phosphorylation of MLKL, the MLKL monomers will assemble into oligomers. So in this diagram, that is what taking place. See, RIPK1 and RIPK3 gets phosphorylated. Okay, so sorry, not RIPK3. MLKL gets phosphorylated from with the help of RIPK1 and RIPK3, and this phosphorylation of MLKL, what will happen is they all will monomers will assemble into oligomers and will move from cytosol to plasma membrane and cause plasma membrane disruption. So this plasma membrane disruption, as we have seen earlier, is a characteristic of necrosis. Okay, so that is what about necroptosis. So this is the diagram is needed to be drawn for necroptosis. So next let us see, okay, before that we will see about some examples in necroptosis. So necroptosis occurs, physiologic necroptosis, necroptosis occurs in both physiological and pathological conditions. In physiologic condition, necroptosis occurs during the formation of the mammalian bone plate. In pathologic sense, it is associated with steatohepatitis, acute pancreatitis, ischemia, reperfusion injury, and neurodegenerative diseases such as Parkinson's disease. Necroptosis also acts as a backup mechanism in host defense against certain viruses that encode caspase inhibitors. Example, cytomegalovirus. Next, let us see what is pyroptosis. As the name indicates, it is a form of apoptosis. It comes under apoptosis and it is accompanied by the release of fever-inducing cytokine IL-1. So the medical term pyro means fever. Since it is associated with the release of fever-inducing cytokine IL-1, the process of apoptosis is known as pyroptosis. And this microbial products which enter infected cells are recognized by cytoplasmic innate immune receptors and can activate multiprotein complex called. So what happens is the activation of multiprotein complex called inflammasome occurs when these microbial products microbial products enter infected cells. So what is inflammasome? This multiprotein complex inflammasome does is it will activate caspase 1. Okay, so it releases a precursor form of interleukin 1 and releases its biologically active form. So IL-1 is a mediator of many aspects of inflammation. Is very important. IL-1 is a mediator of many aspects of inflammation, including leukocyte recruitment and fever. Okay, so that is what it is important in this pyroptosis. So unlike classical apoptosis, this partial cell death is characterized by release of inflammatory mediators, as we have seen earlier. Then pyroptosis is thought to be a mechanism by which some microbes, so there are some microbes which cause the death of infected cells and at the same time trigger local inflammation. So when there is a death of infected cells and local inflammation is triggered, then it is nothing but pyroptosis only. Next, let us see about ferroptosis. So it was discovered in 2012. Ferroptosis is a distinct form of cell death which is activated when excessive intracellular levels, is very important, excessive intracellular levels of iron or reactive oxygen species overwhelm the glutathione dependent antioxidant defenses. It will to cause unchecked membrane lipid peroxidation. So this intense peroxidation takes place. So the intense widespread peroxidation of lipids will disrupt many aspects of functioning of membrane, including fluidity, lipid protein interactions, ion and nutrient transport and signaling pathways. So what happens is there is a loss of plasma membrane permeability, which ultimately causes cell death resembling necrosis. Okay, so this process is however regulated by specific signaling and can be prevented. So ferroptosis can be prevented by reducing iron levels. So the most prominent features of ferroptosis is the loss of mitochondrial cristae and ruptured outer mitochondrial membrane. Okay, next let us see about key constants of necroptosis and pyroptosis. Once we will recall, necroptosis will resemble necrosis morphologically, but like apoptosis is a genetically controlled form of death. That is very important. How necroptosis resembles necrosis morphologically means, we should remember that cell swelling and other processes like loss of ATP, increased permeability of membrane, swelling of the cell, loss of ATP, generation of ROS, reactive oxygen species, and rupture of plasma membrane. This is the characteristics shared by necroptosis and necrosis. So necroptosis is triggered by ligation of TNFR1 by proteins found in RNA and DNA viruses. So phosphorylation of MLKL is very important feature. Okay, how it occurs is because of RIPK1 and RIPK3 signaling, and it will form pores in the plasma membrane. That is the outcome of phosphorylation of MLKL. Okay, so release of cellular contents takes place to evoke an inflammatory reaction. Next, we will receive a pyroptosis. So pyroptosis activates caspase 1 through the help of caspase 1, which will activate the precursor form of IL-1 to generate active IL-1. Caspase 1 along with other closely related caspases also cause death of the infected cell. Ferroptosis is an iron dependent pathway of cell death induced by lipid peroxidation. That is very important. Iron dependent pathway and lipid peroxidation is the key word expected in ferroptosis. Next let us see what is autophagy. Autophagy is a process in which a cell, as the name indicates, a cell eats its own contents. Okay, so we will see it fast. So in autophagy, what happens is the lysosome will the delivery of cytoplasmic materials to the lysosome for degradation. That is what is autophagy. So autophagy is a very important process in which the cells will recycle the digested contents, and autophagy is implicated in many physiological states such as aging and exercise and pathologic process. So formation of phagophore. So phagophore is nothing but the formation of an isolated membrane and also nucleation. This phenomenon occurs only in autophagy, and this two phenomenon is derived from the endoplasmic reticulum, though other membrane sources this plasmid mitochondria may contribute. And then formation of vesicle called autophagosome from the isolation membrane will takes place, and autophagosome will fuse with the lysosomes to deliver digested enzymes to autophagosomes that will degrade its contents. So this diagram you can understand that there is a formation of phagophore. This is phagophore, it's a double membrane layer, and then it elongates and it will join to form the mature autophagosome, and this autophagosome combines with lysosome to form autophagosome. Fusion with lysosome to form autophagosome, and then it will result in recycling of metabolites. So if autophagy related genes are present which will mediate this process, so environmental factors like nutrient deprivation, depletion of growth factors activate an initiation complex of 4 proteins that promotes the hierarchical recruitment of ATGs, that is autophagy related genes, to nucleate the initiation membrane. So as I said earlier, autophagosome is formed by the fusion of initiation membrane of phagophore. So this closure of initiation membrane needs coordinated action of two ubiquitin-like conjugation systems resulting in covalent linkage of lipid phosphatidyl Okay, so this phosphatidyl lipidated LC3 is increased. Phosphatidyl lipidated LC3 is increased during autophagy. It is a useful marker for identifying cells in which autophagy is occurring. So for a cell to find whether autophagy is occurring or not, we need the presence of phosphatidyl lipidated LC3 will show the positive result. Will be increased and present during autophagy. So this autophagosome fuses with lysosome to form an autophagolysosome. So in the last step, the inner membrane and enclosed cytosolic cargoes are degraded by lysosomal enzymes. So that is what about autophagy. Next, autophagy is functions as a survival mechanism under various stress conditions and helps in maintaining the integrity of cells by recycling essential metabolism, clearing intercellular debris. That is important. And then it is more prominent in atrophic cells, degenerating cells, which are exposed to severe nutrient deprivation. So autophagy is also involved in turnover of organelles like endoplasmic reticulum, mitochondria, lysosomes, and clearance of intracellular aggregates that accumulate during aging, stress, and etc. Next, let us see about how autophagy plays an important role in human diseases. So autophagy can promote cancer growth and act as a defense against cancers. That is very important. And neurodegenerative disorders, many neurodegenerative disorders are associated with this regulation of autophagy. Alzheimer's disease, you know, that is characterized by impaired autophagosome maturation. If there is impaired autophagosome maturation, then Alzheimer's disease will take place, and also it is present, and also it is vital, and also it will accelerate neurodegeneration if there is genetic defects in autophagy. In Huntington disease, a mutant gene Huntington impairs autophagy. So autophagy should be present. At the same time, autophagy should not accelerate neurodegeneration. Next, in infectious diseases, many pathogens are destroyed by autophagy, including Mycobacteria, Shigella, and HSV1. So that is helps in engulfing and destroying the infectious pathogens. Inflammatory bowel diseases. So there is a genome-wide associated studies have linked both Crohn's disease and ulcerative colitis to single nucleotide polymorphism SNPs in the autophagy related gene ATG16L1. However, these polymorphisms promote intestinal inflammation is still unknown. So what they're telling is there is a genome-wide association studies that have connection. There is a connection between two diseases as Crohn's disease and ulcerative colitis to single nucleotide polymorphisms in this particular gene. So this defect in this like the single nucleotide polymorphism in this gene can be used to treat both Crohn's disease and ulcerative diseases. These are inflammatory bowel diseases. Next, let us see about mechanisms of cell injury in the next video. Thank you.